Hello. People are joining now. Um, Sorry for being late. For some reason, the what is it? The Canvas page didn't update, and I was in the lecture class waiting for students to log in until I discovered that I was in the wrong class. So, are there any questions before we begin? Let's give a minute or two for some other students to maybe join the class because of um, the problem that I had. All right, let me see. We're going to be discussing lab 7 today. Today is February 12th, and we'll be discussing the throat and urine cultures. Hopefully sometime tonight, I'll send out your unknown project and your first results for the first 3 tests. Any question on any of that? Oh, shoot, not quick enough. Get the exercises up here. Remember when you submit the labs to only submit the exercises, do not submit the lab modules. The lab modules do have the questions in them, but they are not the latest questions. So you may miss some questions if you only answer the questions in the lab module as well. But I do not want the lab module uploaded because it's much bigger. And if every student were to upload the lab module, my Canvas website would soon fill up, and then there would be no way for students to load the assignments. Any question about any of that? All right, so we're covering Lab 7 today. Let me see where we are. Hmm. All right, let's get out of here. And
I did notice something, and I don't know if anyone else did, but on lab 6, question 3 wasn't, it was like in the whole lab file, but it wasn't in just the worksheet file. So I don't know if other people caught that and turned it in as well, but
you're saying it was in the lab module, but not in the worksheet?
yeah, it said using words explain the spreading pattern that you would use to obtain confluent bacterial growth on an agar plate. And so anyway, that wasn't in the worksheet file. It was only in the lab file.
Uh, yeah, you don't need to answer that question.
Oh, well, I did.
Okay, no problem, no
But
penalty.
okay. I didn't want anyone else to get dinged for it.
No, well, thank you, but no, the lab module is not updated and the lab worksheets are, and like I said, there are a few changes and it's only in the worksheet where the change will be. I should pull out the questions from the lab module, and I just haven't had time to do that. Whoever made this wanted the questions in the lab module, and he wanted it in the worksheet as well. And in the first place, now when you update the file, you got to change both files. And in the second place is that I do not want students submitting the lab module. So I'd rather have the questions pulled out, and I just haven't had time to do that yet. And I probably will not do it this term, because if you do not know, I am working over 100% capacity, and that's because the lead for microbiology, he's the one full-timer, and he's not teaching this term, and they gave me all of his classes to teach this term. And I decided to do that because it's only one term, and it'll be one rough term, but next term I'll go back to my usual schedule. Anyways, lab 7, am I sharing my screen? Anyone? Am I sharing
No,
my screen?
no, I don't see it.
All right, let me get that done. Oh, look at that. The lecture file is finished. Let me close that down, and now that will speed up my computer. Here's the, oops, that's the wrong thing. Cancel that. Pushing the button to end the meeting instead of share the screen. Here we go. All right, lab 7, throat and urine cultures. You need to do some pre-reading for this lab. There is some supplemental material, only the video clips. We pulled out the virtual lab because they're using Flash, and Flash is no longer supported and no longer runs. And so we pulled it out of the lab. Let me blow this up a little bit so I can see it a little easier. On the learning objectives, 1, be able to explain how to properly collect throat and urine specimens for routine laboratory culture. 2, list the common bacterial pathogens isolated from the throat and the urine cultures. 3, know the culture media used to cultivate these pathogens. And 4, recognize which tests are used to identify throat and urine pathogens. And then lastly, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will talk about the XV factor testing, but I'm not going to require you to understand it or to explain it, and you will not be tested on it. But you will be tested on the catalase test, the coagulase test, and the oxidase test. So the introduction. Once specimens are received by a laboratory, they are logged and processed in a timely fashion, and specific culture media are inoculated and incubated to grow up the cultures, throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For throat, we mostly grow things on blood agar, but we occasionally grow throat cultures on chocolate agar. Streptococcus pyogenes is the main pathogen isolated from the throat. But we also can find Haemophilus influenzae in babies and young children. Streptococcus pyogenes will grow on blood agar. It will not grow on a general purpose media. You need to have an enriched media like blood agar. Haemophilus influenzae is in the throats and in young children, and it will not grow on blood agar, but it will grow on chocolate agar. And if you do not recall, chocolate agar is similar to blood agar, meaning both have blood added. The difference is blood agar is added to the agar after the agar is cooled to between, I think it's 50 to 55 degrees Celsius. And that keeps the blood from lysing. And once you pull the chocolate agar out of the autoclave, you quickly add the blood while that the agar is close to 121 degrees Celsius. It'll be slightly less, but the point is the agar is very hot. Let me think about that. I think it'll be boiling when you pull it out, so it'll be around 100 degrees Celsius. And that will lyse the blood, and that will make the culture look, the media look brown in color, why they give it the name chocolate agar. Other than that, that's the only difference between the two media, the chocolate agar and the blood agar. But the chocolate agar has the blood cells, red blood cells lysed. The blood agar does not. And Haemophilus influenzae will grow on chocolate agar, but not on blood agar. All right, for the urine specimens, meaning somebody who had a urinary tract infection or some bladder infection, we grow the cultures on first blood agar, and then in addition on either McConkey's or EMB agar. Blood agar will grow all of the urinary tract pathogens, both the gram positives and the gram negatives. Most of the urinary tract infections are caused by a gram negative, specifically the Enterobacteriaceae family. That is the majority of the urinary tract infections. The number one is actually E. coli, the number one urinary tract infective agent. And it grows on McConkey's agar and EMB, and these only grow the gram negatives. And that's why we grow it on two media. We need to grow it on blood agar to grow Streptococcus, Enterococcus, and Staphylococcus. Blood agar will grow Enterobacteriaceae, but to separate out the gram negatives, we also grow it on McConkey's agar and EMB or EMB agar, and that will grow the gram negatives, but not the gram positives. For throat cultures, sore throats, if they're caused by a bacteria, you will see white spots on the throat, on the what do you call it, the tonsils or the back of the throat, as well as white spots on the tongue for a bacterial infection. A viral infection, you will only see red swollen tonsils and red sore throat. You can see the red with the bacteria, but the bacteria have, in addition, the white spots. Most common bacterial pathogen of a sore throat is Streptococcus pyogenes. That's its scientifically named term. It's also called group A strep or gas. I don't use the gas. I do occasionally use the group A streptococcus. And that's just separating the streptococcus by their antigens. Streptococcus pyogenes does cause strep throat. With that, what you do is you swab the back of the throat and the tonsils with a swab. You do not touch the sides of the mouth or the tongue or the roof of the mouth, only the back of the throat and the tonsils. Shown here, it helps if you have a tongue suppressor, hold the tongue down and then swab the back of the throat and the tonsils. Patients that test positive for Streptococcus pyogenes are placed on antibiotic therapy. The reason is that patients generally cannot throw off a Streptococcus pyogenes infection. Streptococcus pyogenes has a capsule around them, and that helps protect the cell from the immune system. Particularly, it does protect the cells from phagocytosis, meaning the white blood cells cannot engulf the Streptococcus pyogenes cells and then destroy them. Since Streptococcus pyogenes is fastidious, it needs to grow on black BAP. That's blood agar plates. You then grow the culture on blood agar and grow it and look at the plate. And if you see small white colonies with a clear region around the colony, and that complete zone of hemolysis called beta hemolysis, that's likely to be Streptococcus pyogenes. There are only 2 tests that are required to prove that it is Streptococcus pyogenes. We run the catalase test and the basotrace susceptibility test. So here we have a blood agar, and that is not Streptococcus pyogenes. It's not showing beta hemolysis. That's actually alpha hemolysis because it's darkening. This plate here shows beta hemolysis. Let me blow it up way, way big. There you can see the dot white colony of bacteria, and it's surrounded by fairly large, clear beta hemolysis. And that is, in this case, it's actually Streptococcus pyogenes, but at the moment, we can only say it is some beta hemolytic colony. The catalase test is then used to show that the bacteria is Streptococcus. Catalase is an enzyme which breaks down hydrogen peroxide into water and oxygen. With the production of oxygen, you then see bubbling. So on this slide, this is catalase negative. No bubbles shown in the hydrogen peroxide. Here is catalase positive. There is bubbles in the hydrogen peroxide. And Streptococcus will be catalase negative. Unfortunately, Enterococcus species are also catalase negative. Most species that grow in the air are catalase positive, like Staphylococcus are catalase positive. Now, Streptococcus does grow in the air, but it is catalase not negative. So does Enterococcus grow in the air. You then run the basotracin test. Basotracin is an antibiotic. So what you do is you take your beta hemolytic bacteria, smear it all over this portion of the blood agar plate. You then put down the basotracin disc, incubate the plate, and if you have a zone of inhibition around the basotracin, and it has to be a specific size, which isn't stated here. I'll look at that. If you have a zone of inhibition that's a specific size, that means that this is caused by Streptococcus pyogenes or group A strep. This bacteria is sensitive to basotracin, and the other members of Streptococcus, as well as other beta hemolytic bacteria, are not sensitive to basotracin, and this is not Streptococcus pyogenes. Any question about how we determine whether we have Streptococcus pyogenes? And any question about the catalase test or the basotracin sensitivity test? All right, another organism that's clinically significant and is isolated from the throat of young children or babies is Haemophilus influenzae. This is a fastidious organism, but it does not grow on blood agar. The reason why Haemophilus influenzae does not grow on blood agar is that Haemophilus influenzae does not lyse the red blood cells the way that Streptococcus pyogenes does. But you can get Haemophilus influenzae to grow on cap, that is chocolate agar, and that's because the blood cells have been lysed, releasing the factors that Haemophilus influenzae requires. You can get Haemophilus influenzae to grow on blood agar if you grow Staphylococcus aureus on the blood agar and then streak the Haemophilus influenzae through the Staphylococcus aureus. So what you do is you have blood agar, and then you put a single streak of Staphylococcus aureus down the blood agar right here. And that's what that thick line is. Let me blow this up. And then you streak the Haemophilus influenzae in this region through the streak of the Staphylococcus aureus. And if you can notice, the Haemophilus influenzae is not growing in this region of the streak, but it is growing around the streak of Staphylococcus aureus. The reason why the Haemophilus influenzae can grow in this region of the streak is that the Staphylococcus aureus lyses the red blood cells, and then the nutrients that are required by Haemophilus influenzae will diffuse from the lysed red blood cells in this region of the plate. And so Haemophilus influenzae does grow in this region of the plate around the Staphylococcus aureus, but it will not grow in this region where we have it streaked because Haemophilus influenzae needs some nutrients from the blood, and Haemophilus influenzae cannot lyse the red blood cells to release the nutrients. Any question about any of that? The factors released from the blood are the X and the V factors, and Staphylococcus aureus will lyse the blood and then release those factors, allowing the Haemophilus influenzae to grow. However, because the red blood cells are lysed in the chocolate agar, the X and V factors are released, and these two factors are required in the presence of the media in order for Haemophilus influenzae to grow. All right, any question about any of that? All right, let's move on to urine cultures. They are used to diagnose a urinary tract infection. Most of the time, that's your arthritis or cystitis, infection of the bladder, but it can be polynephritis, and that's infection above the bladder, either the ureters or the kidneys. If you don't know if you have a bladder infection that goes all the way up to your kidneys, that is a very serious disease. My mother actually had that when she was quite elderly, and that was a very serious infection. You do not want your kidneys infected. It's bad enough to have your bladder infected, bad enough even to have your urethra infected. But frequently the infection, if it is a mobile bacteria, will just crawl up from the ureters, the urethra, infect the bladder, crawl up from the bladder to infect the ureters, and can crawl up the ureters to actually infect the kidneys. Females are more predisposed to developing a UTI than a male, and that's simply because they have a shorter urethra. To sample the urine to determine whether the patient has a urinary tract infection, the genital area must be clean prior to collection, and then you obtain a clean midstream collection where you start voiding the urine, and then in the middle of the urine you collect it for the clean midstream. This is the way patients prefer to give it. There is some bacteria that will come from the urethra and then contaminate the urine, meaning the urethra does have bacteria in it, and we generally just do the clean midstream, and the patient prefers that, but you can put a catheter up the urethra and then sample the bladder. However, that itself can produce contamination because the urethra has bacteria in it, and you can, with the catheter, pick up the bacteria and then put it in the bladder and then sample that. And so even the catheterization is not bacteria free. To get bacteria free, you have to use a suprapubic aspiration, put a needle directly into the bladder, and then sample the urine directly from the bladder. Patients especially don't like this because that's a puncture wound into their bladder, and obviously you have to get a needle stick. But if you want a bacteria-free sample and directly sample the urine from the bladder, you need to do that. Now, let me state that even that will not be bacteria-free. At least among females, they do have a normal microbiota of bacteria living in the bladder. I don't know if males have a normal microbiota in the bladder because all of the studies have only looked at females. So I don't know if males have a normal microbiota in the bladder, but at least females do. And so you can get the normal microbiota directly from the bladder, even in the suprapubic aspiration. But at least you're not getting the contamination from the urethra. Anyways, those are the 3 ways to get a urinary tract sample. The clean catch midstream, the most common way. You can get it from a catheter, and you can get it from suprapubic aspiration. Once the urine is received in the lab, 0.001 millimeter, that's 1 microliter of urine, is placed onto a non-selective media like blood agar with an inoculating loop, which actually measures out that volume directly. I've got a picture of it right there. Let me blow this up. Right here. So this loop will pick up 0.001 milliliter, that's 1 microliter, and then you put it on the blood agar plate like that, then streak it to get the growth like that. In addition to growing the bacteria on BAP, we also grow the bacteria either on EMB or McConkey's, and that's to grow up the gram-negatives, especially the Enterobacteriaceae family is the most likely family to cause a urinary tract infection. The number 1 cause of a urinary tract infection is actually E. coli. Let me shrink that down and go back to where I was. Coliforms, especially the Enterobacteriaceae, are the most common isolate. isolated from the urinary tract infection. Coliforms are just a bacteria that grows in the colon. Most of them are Enterobacteriaceae, members of that family. Interpretation of a positive urine culture is multifaceted. Factors that need to be considered is the type of collection, voided versus catheterization or the suprapubic aspiration. Obviously, the more invasive you are, the less contaminating bacteria you'll pick up. The gender of the patient. For some reason, females have more bacteria, at least in the voided urine. They may have more bacteria in their bladder, in which case they have more bacteria for that reason as well, than male patients. The number of colonies per mil per urine, take a look at that. And then the presence of polymicrobial growth. If multiple organisms are isolated, it may indicate a poor collection technique. Like the patient, let's assume they're using a clean midstream sample. They may have poorly cleaned their genitals before collecting the urine. All right, so then once we have the bacteria growing on either the blood agar or the McConkey's agar or both, we then do some biochemical tests to try and identify the species. We take a look at the presence of coagulase. This enzyme is found in Staphylococcus aureus, and Staphylococcus aureus has coagulase. The other members of the Staphylococcus species are coagulase negative. Coagulase production contributes to the pathogenicity of Staphylococcus aureus. Coagulase converts the plasma into a blood clot. And what happens is coagulase acts on the fibrinogen to make the fibrin clot fibers, and then that will make a clot. The clot will protect the bacteria when the bacteria are few in numbers. So Staphylococcus aureus, when it first gets established in a host, it produces coagulase, which then produces a clot around the Staphylococcus aureus, and the clot makes it harder for the immune system to attack the Staphylococcus aureus. When the Staphylococcus aureus is first getting started and is few in numbers. And that helps protect the bacteria. What happens is the bacteria have to eat through the fibrin clot before they can get to the bacteria. Did I say the bacteria? I mean the red blood cells have to digest the fibrin clot before they can get to the bacteria to attack the bacteria. The next test we're going to look, this is the coagulase test, sorry. We have two tests. Whenever I'm doing the unknown test, I'll give you the tube test where you have a clot and the plasma clumps. And this is the plasma as a liquid and it doesn't clump. That's coagulase negative, coagulase positive. You can do the slide test, coagulase negative. The plasma is clear and smooth. The coagulase positive, you'll see clumping. All right, the oxidase test is a test we use to separate out the Pseudomonaceae family from the Enterobacteriaceae family. The Pseudomonaceae are oxidase positive, the Enterobacteriaceae are oxidase negative. Both the Pseudomonas and the Enterobacteriaceae can cause urinary tract infections. Both the Pseudomonas and the Enterobacteriaceae are gram-negative bacillus. They do have some differences besides the oxidase test. The Pseudomonas are perform aerobic respiration. The Enterobacteriaceae are facultative anaerobes. They will perform aerobic respiration if oxygen is present, but if oxygen is not present, they perform anaerobic respiration or fermentation. The Pseudomonaceae do not ferment the sugar glucose. The Enterobacteriaceae do ferment the sugar glucose. And the easiest test to do is the oxidase test because you just look at it, and oxidase is looking at the presence of cytochrome oxidase C, which is an important enzyme in aerobic respiration. And the Pseudomonas have it. And so they will be oxidase positive, and you'll see a bluish or purplish color on the oxidase positive cells in this region receive the oxidase test. The Enterobacteriaceae will not be oxidase positive. They will not develop the blue or purple color. Any question about any of that? All right, is that the three tests I talked about? Oxidase, coagulase. I talked about catalase earlier, because you'll need to know about catalase as well. All right, you can skip the references. Surprisingly, we don't have terms for this one. You're not going to be physically performing any of these activities. However, you will be responsible for knowing how to perform them. We have a little video on viewing the catalase test, so watch that video. We have a little video on performing the coagulase test, so watch that video. And then we have a video on performing the oxidase test, so watch that video. And then let's go to the worksheet. Answer the questions. The first couple of questions are going to talk about the enzymes catalase, coagulase, and oxidase. Which enzyme converts fibrinogen into fibrin? And name an organism that tests positive for this enzyme. So 1A is the enzyme, 1B will be an organism that tests positive for this enzyme. 2A detects, what enzyme detects the presence of cytochrome C proteins in the electron transport system? And 2B, to name two organisms that test positive for this enzyme. They're mentioned in the video clip. I believe I mentioned them also in the lab module. 3, which enzyme hydrolyzes hydrogen peroxide into the non-toxic products water and oxygen? And name two organisms that test negative for this enzyme. 4, which rapid test is used to differentiate between the following? Between 4A, Staphylococcus and Streptococcus. And by rapid test, we're talking about one of these three enzymes. And then 4B, which rapid test can distinguish between Staphylococcus aureus and Staphylococcus, other Staphylococcus species. And then 4C, which rapid test can distinguish between Enterobacteriaceae, such as E. coli, and Pseudomonaceae, such as Pseudomonas aeruginosa. 5, Concerning specimen collection for routine throat culture, what areas of the throat should be swabbed? And 5B, what areas of the mouth should be avoided? 6, what is the most common bacterial cause of pharyngitis, meaning what is the most common cause of a bacterial sore throat? And you do need to state a bacteria. So don't give me like the common cold, because that's not a bacteria. 7, what test is used by microbiologists to differentiate between beta hemolytic group A streptococcus and other beta hemolytic streptococcus species isolated in the throat? Remember, group A streptococcus is Streptococcus pyogenes. How do we separate Streptococcus pyogenes from other members of the Streptococcus family? And there's one test. Okay. 8, answer the following concerning a child's throat culture. 8A, state 2 non-selective agar plates that are inoculated for a child's sore throat. And when you're swabbing, you're growing the bacteria. And 8B, what organism can be isolated on CAP, that's chocolate agar, but not on BAP, that's blood agar. So name the organism. 8C, you do not need to answer because you need to do some online research to answer that. So I just scratched it out. 9, list 3 methods that are used to collect urine for routine culture. 10, list 3 reasons why urine specimen should be rejected for culture. You can get that from the lab module. 11, how much urine is placed on the surface of a blood agar plate? 12, what is the most common bacteria isolated from patients with cystitis, and that is cystitis, and that is a urinary tract infection. 13, identify the gram reaction and the cell morphology, meaning give me the gram stain and the shape for these bacteria. If you cannot find the answer in the module, you need to do some research. Most of them are in the module, but this module, I forgot to mention that, has a supplement at the bottom of it. And so give me the gram stain for Staphylococcus saprophyticus and the cell morphology. And then for E. coli and Pseudomonas aeruginosa, Streptococcus pyogenes, and Haemophilus influenzae. And Haemophilus influenzae is mentioned in the lab module at the bottom of it. So let's go to the appendix. It talks about the streptococcus in the appendix. It talks about the beta hemolytic streptococci. And then it talks about Haemophilus, specifically Haemophilus influenzae in the appendix. Oh, and it does talk a little bit about coagulase, that enzyme. I must have put that because I thought that there wasn't quite enough information in the lab module. All right, any questions about this? This is one of the easier labs, meaning it shouldn't take you too long to finish it. There are no case studies in this lab. Let me make sure that that's correct. Yeah, there's only 13 questions. I think all of the other labs from this one, all the later labs, will have case studies, and those will take you a little longer to do. But I will be here until 8 o'clock to answer any questions or until the last student has logged out. And if you're a student who has trouble in the lab and you've gotten a lot of questions wrong, I do advise you to make use of me because I'm here to help you. All right, any questions? If not, go ahead and get started in the lab. I will be here until the last student logs out or until 8 o'clock.